So in this latest video series, we are investigating whether vaccines could be shed, or vaccine material in the form of the spike protein produced from vaccines could be shed. And we're about to start the third installment of this series. In the first installment, we discussed whether what the definition of shedding is and whether material could be shed via breathing. So that's what we talked about. In the second series, we talked about what can we learn about exosomes in vaccinated individuals, because of the fact that exosomes, which are fragments of cells, can be breathed out, and these fragments of cells can carry biological material embedded in it straight from the cell surface. So that could include spike proteins. And we are going to finally commence the third segment. And in this third segment, we're going to be discussing what can we learn about these exosomes, or these small cellular fragments released by cells. And these fragments can circulate or be breathed out, as I mentioned. And what can we learn about these from COVID-19 patients? So today I'm going to present 3 papers for you. So let's get going. My name is Dr. Mikołaj Raszek of Neurogenomics, and let's get started. So in the first paper, I want to tell you, I'm going to start with the conclusion, because few of you have been asking about that. Let me summarize the paper. And basically, in this particular paper, what the authors did is they investigated whether spike protein or the genetic material of the virus is being present to different degree in long COVID patients versus COVID-19 patients with severe disease versus controls. And the conclusion was that long COVID patients have higher levels of spike protein present. All right, so let's summarize that in greater detail. So as I mentioned, they looked at these 3 different types of patients. They looked at hospitalized patients, so those who had acute or severe COVID-19, and they looked at long COVID patients, and then controls, which were healthy individuals. And they measured their levels of spike protein in the blood, as well as the genetic material. And they were able to indeed discover there were spike protein circulating for a long time. And the summary is that in hospitalized patients, those who had spike protein, the spike protein levels actually correlated with the length of hospitalization, as well as D-dimer levels. Now, what are D-dimer levels? D-dimers is basically a hallmark of coagulation taking place, or basically clot formation. And in essence, basically it's a subcomponent of clot processing. And D-dimers levels have been known to correlate with the severity of COVID-19 disease. So the higher the levels of D-dimer levels, the more severe typically the COVID-19 outcomes were. So once again, the conclusion was that those hospitalized with severe COVID-19, circulating spike protein levels correlated with the D-dimers and the stay of hospital stay. Now, they also looked at that in terms of those with long COVID, which is also officially that's referred to as post-acute sequelae of COVID-19, or PASC, as I like to call it. Sequelae basically means complications, so disease complications, in this case COVID-19 complications, colloquial term, long COVID. And again, spike protein levels correlated with D-dimers as well. And drop my ski pole. But what is also interesting is the levels of circulating spike proteins that they were discovering in these individuals, is because they were detecting nanogram amounts per milliliter of blood. That's order of magnitude higher than what was previously reported and that we have discussed in our prior videos, which was in picogram amounts. So basically we're talking about 1,000-fold higher levels of spike protein than whatever has been previously reported in infected individuals or vaccinated individuals. So kind of interesting to see that. But these authors also noted that the spike proteins were often found inside extracellular vesicles. What are these? Those are basically these fragments of cells that are being released. And let me show you some of the background. Fragments of cells being released, they can be different sizes. So the smaller extracellular vesicles, these are referred to as exosomes. So this is what we've been talking about in the last few videos, because exosomes are basically the size of coronavirus. So just to give you a perspective. And then they can be extracellular vesicles, can be larger as well. And they studied both the small ones and large ones. And the spike proteins was being discovered in all of these sizes of extracellular vesicles. And again, presence of spike protein in extracellular vesicles correlated with long COVID. So definitely an interesting observation right there. Okay, so, but there's one more thing that these authors showed that I thought was super interesting is they wanted to see, well, how does the spike protein end up in these exosomes or extracellular vesicles? Well, clearly, if a cell releases a fragment of itself, then that fragment is made out of cell membrane from which that extracellular vesicle or exosome is being released. So whatever happens to be on the membrane of that cell can be captured on these exosomes. So as a matter of fact, then exosomes can act as a marker of what is being present on a cell, and you can actually learn from what the cell looks like, right? So what that means is the spike protein itself could have been on the cell surface, and that's how it ended up in the exosomes. But there's one more thing that could have happened, and that is, it's been previously reported that spike protein can bind to, don't mind the hair, I'm skiing up steep slope, that spike protein can bind to heparan sulfate. So what are these? We talked about that in one of our prior videos. So what is heparan sulfates? It's basically towers of special sugar molecules. These can be found on cell surface. Heparan sulfate is found in all animal tissues, and apparently spike protein can bind to it. So what these authors wanted to see is, could the spike protein also be found on these exosomes or larger size extracellular vesicles, because it interacts with heparan sulfate. And what they did is they took those exosomes isolated from COVID-19 patients, and they treated them with this enzyme that would destroy heparan sulfate. So basically what they were doing is, that enzyme, I think it was called heparinase 2. I might have screwed up the name. But basically the point is that enzyme destroyed that heparan sulfate sugar molecules on a cell surface. And when they did that, they were able to show that indeed the level of spike protein on these exosomes decreased. So this suggests that there might be another way how spike protein could be attaching to exosomes, which is simply by interaction with these sugar molecules bound on the surface of the exosome. So potentially two different ways how spike protein could be moved around by these exosomes. Now remember, one thing that we still want to find out is with the exosomes carrying spike protein could be breathed out. We know that exosomes could be breathed out, but what has never been studied yet is whether exosomes with spike protein could be breathed out. And that's still a mystery. But basically what we're seeing is that exosomes can be carrying spike protein, and they do correlate with disease outcomes and disease severity. So that's basically the conclusion of that study. So I thought that was fairly interesting. But the same authors previously have also shown, so their conclusion was like, look, exosomes with spike protein could be contributing to the severity of COVID-19 disease, including long COVID. But the same authors also shown previously that exosomes isolated from COVID-19 patients could also be leading to cell destruction inside lung tissues. So let's talk about that a little bit. So clearly this was not their first foray into this topic. So in the second paper, again, let's start with the conclusion as requested. Oh man, okay. Here goes the pole again and the hair. Listen, I think I need to give up on the hair. Is that better? All right. So the conclusion was that exosomes of COVID-19 patients could be contributing to the severity of the disease. And in this particular study, what the authors wanted to investigate was, hey, what is the protein cargo in the exosomes, or I should say extracellular vesicles, isolated from COVID-19 patients with different severity of the disease, as well as healthy controls. And basically, when I mean protein cargo, they just wanted to see what kind of proteins are being found. And again, remember, these proteins are being captured from the cells that are releasing these exosomes. So this can tell you some of the information about, hey, what are the cells of these individuals with different severity look like? And the take-home message here is that the proteins that were discovered in COVID-19 patients correlated with being involved in inflammation, being involved in coagulation, or basically creating clots, and basically stimulating the immune system and tissue reorganization. Basically, cells undergo transformation, so they go from one state to another. We're going to talk about some of these proteins in a moment. And but what's really interesting is that you could literally, this is so specific that you could correlate not only whether the person, specific protein type, whether the person had COVID-19 or not, but even the severity of the disease was reflected by specific proteins on the exosomes released by these patients. So very, very interesting information. Not again, I keep dropping my pole. So I think I'm gonna pause right here and calm down. So it's such a beautiful and calm day here. So excellent. So that was super interesting. But what they also then showed is they were curious whether these exosomes isolated from these COVID-19 patients, whether they can infect lung endothelial cells. So endothelial cells are the type of cells that actually make up the walls of the blood vessels, and they did really cool immunofluorescence staining images, and they were able to show that these exosomes are from COVID-19 patients can be taken up by these endothelial cells, by these specific type of cells, lung cells. So what are the immunofluorescence images? We've shown quite a few of these before, including especially in some of the discussion in relation to spike protein potentially entering nucleus. Basically use antibodies that have fluorescent molecules, and of course antibodies recognize a specific protein of interest, right? So you can use antibodies to bind a specific target you want, specific protein you want, and because that antibody is also attached to a fluorescent molecule of specific color, you can then colorize fragments of the cell and know what is where physically inside the cell. So in a previous video, I was able to show you through this type of experiments of immunofluorescence experiments that, for example, how dendritic antigen presenting cells can take up exosomes with as well. And in this case, what the authors showed is they marked these extracellular vesicles, such as exosomes, with specific fluorescent marker, which was either green or red, and they were able to show that these endothelial cells were able to take up these exosomes because they were able to show that when these endothelial cells were exposed to exosomes, they entered the cells. So in one of the image, they were able to show these exosomes fluorescing green because they had a marker with green fluorescence on it, entering endothelial cells, and they marked the endothelial cells in couple ways. They used red fluorescence to show where actin filaments are. And what are actin filaments? Basically, they form the cytoskeleton of the cell. So basically, like the name suggests, it's literally it forms a skeleton inside the cell that gives the shape to the cell and a network highways through which different proteins can move around inside the cell and from one area of the cell to another. So basically this network of actin filament is all over the cell, so you can clearly see that. And then the last staining they used was what is referred to as DAPI staining, and it's bright blue fluorescence, and that DAPI actually goes inside the nucleus, goes inside the DNA of the nucleus, and it indicates where the genetic nuclear material is. Basically, it shows you where the nucleus inside the cell is. So they were able to show that. They also show, and what they did is they treated the cells with an inhibitor of exosome uptake called dinosaur. Yeah, I'm not making that name up. It's just spelled differently. And they were able to show when they were that compound prevents exosomes from being able to be taken up by cells, and when they expose the endothelial cells to that particular compound, these fluorescent extracellular vesicles from COVID-19 patients were either not taken up or severely, or the taking up of these extracellular vesicles severely reduced. They also showed this with red staining, where they stained the extracellular vesicles in red, and the same thing, they were able to show that cells, endothelial lung cells, they were taking up these exosomes that were fluorescing now in red, and again, the nuclei of the lung endothelial cells were fluorescing in blue. So, and then again, when they used the inhibitor, dinosaur inhibitor, that prevented the take-up of the exosome. So clearly, the lung cells can take up these, specific lung cells can take up these extracellular vesicles. But why is that important? Is because more importantly, also these authors were able to show that upon taking up of the exosomes, these cells experience apoptosis. So what does that mean? Basically, it means death of the cell. So the cell undergoes program death, and it obviously kills the cell. So that's an important observation. So clearly, what that means is that exosomes from COVID-19 patients with severe COVID-19 can be dangerous. So that's what we're learning. They did not look at spike protein, presence of spike protein in this particular study, but they did assess other proteins. So let's get into that component. And first, before I start on that, I wanted to also let you know that another observation that they're able to show is that these patients with severe COVID-19, they also experience lymphopenia. That's basically a severe reduction of white blood cells. And they were able to show that one of the proteins in these extracellular vesicles isolated from COVID-19 had a T-cell exhaustion marker called PD-1. And we talked about that before. And we discussed that in one of the videos about what is T-cell exhaustion. Basically, T-cells are no longer responding properly, and they can have a specific marker of that. That's the one. And so clearly that might help to explain why we're observing this this reduction of white blood cells. And now let's get going to some of the other proteins they found. Now, I only want to mention a few. They discussed many. So one of them was called tissue factor, and tissue factor protein is involved in formation of clots. We'll be discussing that in future videos of how clotting occurs in COVID-19 and how spike protein can promote formation of clots. So stay tuned for those videos. But might as well make a little brief introduction here of this content. And basically, the exosomes from severe COVID-19 patients had an increased levels of tissue factor protein, and that has been previously observed as well. That exosomes with tissue factor protein can indeed promote clot formation and fibrin polymerization. Basically, that's how clots are being formed. We'll be discussing that in great detail in the future. And so also they were able to show another protein. I'll mention just a couple more. Another one was they were able to show that a protein called, what I will call it, ADAMTS, I believe 13. That's a protease, meaning it's an enzyme or a protein that destroys other proteins, and this one particularly destroys an inflammatory factor called von Willebrand factor. It's an inflammatory protein that we discussed previously as well. And basically, exosomes, or extracellular vesicles, because they had some exosomes and larger ones, and these had lower levels of those proteins compared to, say, controls, healthy individuals. On the other hand, they had other increased levels of that von Willebrand factor inflammatory marker as well, and many others. But I just wanted to focus on this just because I will be coming back to that when I will talk about clotting in relation to spike protein. So the conclusion there basically was, again, that the extracellular vesicles can promote the severity of the disease of COVID-19 disease. So I want to now mention one more paper, and in this particular paper, what the authors did is, again, they looked at the extracellular vesicles, including exosomes. And of course, in the previous paper, once again, I forgot to mention, the authors definitely also showed the exosomes. They showed cool electron microscopy photographs of exosomes as well. We showed you a few of these before, so here's some more that they were able to show you, and they were able to show the distribution of both the small ones, which are the exosomes, as well as the larger ones. Okay, so in case you didn't have enough images of what these little cell fragments look like. And so now let's come back to that last paper. And in the last paper, what the authors did is they wanted to see exosomes from, again, COVID-19 severe patients. And what they were able to show is that these exosomes, when exposed to, once again, endothelial cells, but they were exposing these extracellular vesicles to endothelial cells from liver, as well as blood vessels and microvasculature. And basically, what they were able to show is that upon exposure to these severe COVID-19 extracellular vesicles, these cells induced what is referred to as inflammasome. What was the name? I think it was NLRP3. I could have messed that up as well. This is from memory, so forgive me. But basically, what is an inflammasome? It's a multi-protein complex. So these are like big complexes, and basically they act like a little sensors. So they sense the outside environment, and based on how they sense it, they can induce inflammation and promote changes to the cellular activity, so that the cell can take certain antiviral actions. So, and they were able to show that basically these inflammasomes are being induced. Now, inflammasomes, these type of inflammasomes have been previously observed in COVID-19 disease, and I believe it correlated with the severity of the disease. So clearly this indicates that, once again, exosomes from or extracellular vesicles of different sizes could be promoting the disease. Now, the authors also mention that SARS-CoV-2 virus itself has previously been reported to induce formation of inflammasomes as well. And the authors wanted to see, hey, if could spike protein induce these itself? So they actually expressed or basically induced certain cells to produce spike protein, and but they were not able to show that such treatment or manipulation was able to produce formation of inflammasomes. But the overall take-home message here is that these extracellular vesicles of either small size, which are the exosomes, or larger size, they can be fairly dangerous in terms of progression of the severity of disease, COVID-19 disease, and its negative outcomes, as well as it might be one of the contributing roles out of many towards what we refer to as long COVID basically. So that's all I'm gonna have for you today. I hope you're okay with the hairstyle, and I look forward to some of you in the new upcoming COVID-19 Q&A. And once again, thank you everyone for your support. Thanks also everyone for the donations. Just wanted to let you know these are deeply appreciated because they help a lot. Why? Because they didn't go into my pocket. They go in the pocket of other people, the people involved behind the scenes that I pay in helping me produce these videos. So I wanted to say big thank you. It's every dollar matters, and it's deeply appreciated. I know I don't say it enough. I'll try to say it more often. It's definitely valued. So thank you once again. And thanks for sharing the videos. Thanks for helping me grow the channel. And thanks for steering the science. A lot of the science is also based on what you're asking, what type of questions are being asked, both on the comments on the YouTube channel, as well as the comments in the COVID-19 Q&A sessions. And I look forward to future installments. Bye everyone for now. Oh, and before I say actually I should say, if you can help it, go outdoors, spend as much time outdoors as you can. If you haven't figured it out, clearly, that's the primary message that I'm trying to drive across in my educational channel. Bye everyone, ciao for now.